Okay, good morning. So thank you all for connecting today for the Apple Forum. My name is Sergey Nachimowicz. Just a little bit of a housekeeping before we really get into it. Please mute your microphones unless you have a question that you wish to ask. It's completely fine for you to interrupt me during the talk. And you can do that, no problem, and ask the question. So if you can, please mute your microphones. That would be great. I would appreciate that so that we don't have any background noise. And we're recording this meeting just for you to be sure what's happening here. And we're going to go and start it off. So my name is Sergey Nachimowicz. I'm a plant pathologist at the Hudson Valley Research Lab. This is my 5th season at the lab, and I have been serving the industry here in the last 5 seasons. And I will be talking about the overview of the plant pathology program and the achievements we had in 2020. Thank you so much for connecting. I know there's a lot of Zoom meetings, but I hope that this meeting is going to be memorized pretty well in your time. Please mute your microphones if you can, so that we can actually have a good sound and no interference from the back.
And for Biden's circumstance at trying to push his foreign.
Okay, thank you. So today I will cover 4 sections of this talk. First of all, I'll talk about the Colototricum species and their identification, which are the behind the group of species that cause bitter rot in apples. And we're going to talk about the Mid-Atlantic region, including primarily New York, Pennsylvania, and Virginia. And we're going to talk about distribution, habitat, and management of the different diseases, different of these species in terms of the fungicides. Now we'll segue into the second section, which is going to be on a viability digital PCR, which is basically a molecular identification method that is basically helping us do research on fire blight pathogen. And then I will segue into the Marsanina leaf blotch or apple blotch disease, which is caused by Diplocarpon coronaria or Marsanina coronaria. And then close with the last section on the efficacy trials done at the lab in the 2020. So in the first section, talking about the Colototricum species, I'm showing you here different symptoms of bitter rot that can vary from region and also from different cultivars. And, this, which led us to hypothesize that because of the slightly different symptoms that we might expect to see also different species, and that some species might be unique in their presence and prevalence in certain regions. So this problem has not been really a new problem. It has been known in the region that from late 1900s, late 1800s into early 1900s, the first written records talk about Colototricum species, or actually bitter rot as a disease. But however, we didn't know which species is dominant in New York, and we didn't know which of these species that are known in literature cause bitter rot in New York. And we did have issues with bitter rot in 2015 and 16. There was incidence of up to 5 to 7% of storage, spots that were caused by bitter rot. We also heard from our colleagues in Canada that we had issues with bitter rot there in Ontario region. And overall, in certain years, such as 2017, 18, we have seen losses up to 60 to 80% in certain orchards. However, we did not have severe outbreaks in 2019 and 20. In 2020, likely because of the drought, which was most likely the reason why we did not see too much problems with this disease. But we did have in 2016, 17, and 18. So this is the life cycle of of all the Colototricum species. You can see this larger blue circle there. That is a sexual reproduction stage of this fungus, which allows formation of perithecia, which is very similar to the apple scab fungus. In the perithecia, the pathogen forms ascospores, which are windborne and can reach the leaves and infect also fruit and leaves. However, we are not sure whether this stage, sexual stage, plays an important role in infection by these fungi, because It has been described in literature that it doesn't occur in the northern regions that often, but mostly in the southeastern United States. The inner circle is the life cycle basically continuing in the asexual reproduction stage. Pathogen survives as mycelium in the branches and cankers, and also it can survive on the fruit mummies that are present in the canopy and also on the ground floor, on the floor of the orchard. And in spring, the pathogen forms a ceruli, basically a saucer flat-like bodies in which conidia are formed, and conidia are rain splashed. They're not as mobile as ascospores are. They require rain, except in the cases when there's violent wind that actually carries aerosol and carries spores with it. Once the pathogen infects, it has the ability to go into a dormant state or quiescent latent period. And that's why we call this pathogen a hemibiotroph, meaning the infection is established, the symptoms are not occurring, and pathogen is basically dormant. And that's called a hemibiotroph. And then after a certain amount of time, when the certain chemical cues trigger the pathogen, or environmental cues trigger the pathogen to continue infection, we can get rots occurring on the fruit and also on different other organs or on leaves, and then we see the rots. On top of that ability to be a hemibiotroph and a necrotroph at the same time in a lifestyle of this pathogen, the pathogen can also dwell inside of the leaves of the host and other non-host plants in the orchard, such as weeds, and also around the orchard, such as forest trees. And it can live there freely, produce spores, and spread, and also be exposed to fungicides during all this time, which is very important in terms of the resistance of these fungi to the fungicides. So why do we have more bitter rot in recent years? Well, we did have the case where we know that implementation of 77 days pre-harvest interval for mancozeb, which is very effective fungicide for these fungi, has kind of maybe contributed to the larger incidence. We do have more hotter and humid summers, and we do grow and plant very vigorously multiple varieties that are very susceptible, and of course, market demands them. And also we do see a lot of fire blight, which allows this pathogen to move onto the symptoms of fire blight, and then survive better on the cankers and branches. We also see there's no orchard sanitation in many cases when the bitter rot occurs. And widely through the region, we see that late maturing cultivars can have more bitter rot because likely the fungicide residues are not being maintained up until harvest. And that actually is very important. However, we do see that north of Albany, there's just less problems with this disease, except in some pockets close to the Champlain Lake Valley and also close to the Ontario region. So we do see sometimes problems with it, but not as frequently as we see it in Hudson Valley region. There is an implication that we see more bitter rot in drought-stressed trees, that we see more bitter rot when there is fruit injury associated with insects and also with different environmental factors such as hail. If there's no fungicide coverage, the pathogen can spread rapidly, and these small lesions very often occur near harvest, and fruit are harvested and then they express in the storage. And we have seen that in certain cases, and the symptoms can be very similar to other rots. And that, therefore, the diagnostics of the problem is very important, both in the orchard and the storage. And we have many questions, of course, whether these fungi, when do they form this dormant infection? Which factors contribute to the infection, such as heat injury? How successfully these pathogens survive in buds or different other organs of the plant? And of course, we didn't know which fungi we have in the Hudson Valley to cause this disease, but many other questions are really burning. So if you look at the overview of different species, this is the literature basically stating that in these three groups that you see here, the one that I highlighted in blue font, red font, and white font, these three species complexes, that's where we see the most of the species that infect apple belong to. The first species complex is called Colotatricum nicotarium species complex, and these species dwell more in the cooler regions with temperature optimums around 77 Fahrenheit. And the ones that I highlighted in yellow font, those are the species that have been reported so far in the United States to cause bitter rot on apple. And the second group is Coleotatricum gliosporidae species complex, which belong to and dwell more in warmer regions of tropics and subtropical regions with temperature optimums around 86 Fahrenheit. And the yellow species one that I highlighted were reported so far in the United States to infect palm fruit. You can see that this group in red actually can also cause symptoms on leaves. And if you go to North Carolina and southern part of Virginia and South Carolina, you can see actually a leaf form of this disease called glomerular leaf spot. And it's different from the leaf blotch or physiological disorder on the golden delicious and other similar varieties, because it has concentric rings in the middle of these spots. However, we rarely see that in New York. And the third complex, we don't see, even though we don't see this last species in United States, but it is described in the world as is causing the bitter rot. So we collected roughly 500 isolates from 14 different locations in New York, primarily in the Hudson Valley region. And this is how the isolates look like. So in the first row, you can see this deep pink color of the isolates, and 60% of them looked like that. On the underside, it has this deep maroon color. And this description of the morphology of the fungi belonged to the Coleotatricum acutatum species complex overall, and that's what the CAS stands for. And then we had, in the second row, you can see gray, white colonies that took about 25% of the isolates that we collected. And then we had completely white isolates at the bottom that, basically had 12.5% of the isolates. In the last two rows, gray, white colonies and white colonies of these fungi really resembled in their description to the Coleotatricum gliosporidae species complex, but we were not sure what these species are. So what we did is we conducted a phylogeny. Basically, we sequenced certain fingerprint regions in the DNA of these fungi. And basically what you see here is a family tree that shows evolutionary relationships between reference strains that are uploaded to a large database of NCBI that allows us to blast or match these fingerprint DNA regions we sequenced to those reference strains. And what you can see here, the first question we answered was whether the isolates we collected belong to two complexes. And on the left side, you can see all the isolates that have AFK label. Those are the ones from New York and Virginia and Pennsylvania. And they belong definitely to gliosporidae species complex. So that definitely means that we do have the species from the warmer regions present in the Hudson Valley. And then on the right side, you can see that ACF K isolates, they're from New York and other regions that they belong to also acutatum species complex. So we do have both groups of species from warmer and the cooler regions. So that was the first question that we answered. And then the next question we had was that 60% of the isolates, based on the gene sequencing of three specific gene regions or fingerprint regions in this phylogeny tree, you can see that in this family tree, that those species basically were identified as Coleotatricum fierineae, so 60% of the isolates. And why is this important? Well, it is important because this species, is widely present, and it belongs to the same group or phylum where the Venturion equalis belongs, or the apple scab fungus. So this is one of the sisters of this species. It is widely present throughout the world as a pathogen of vegetables and fruits. There's over 100 different plant species worldwide that get infected by this species, and it causes bitter rot on apple. And also it has these variable lifestyles that I mentioned to you before. And you can see here that in this family tree, there's many different clades or groups, and these other species which are sister species to Coleotatricum fierineae. But this is by far the most dominant species. And then we teamed up with Carrie Peters' lab at Penn State and worked with Philip Martin, her student, who actually collected Canidia with spore traps and wanted to compare what is the amount of spores that he's going to collect in the forests where we know likely this pathogen dwells as an endophyte, just freely living, not causing any disease, just living inside or on the surface of the host. And he quantified Canidia, and you can see here in these whisked bars that overall, that Conidia had much higher presence in the woodlots, basically around the orchards, meaning that most likely the primary lifestyle of this fungus is an endophyte, even though it is present in the orchards. And you can see here when the fungicides are applied in the orchards, you see a lower incidence of the spores, but overall, it seems that the larger, more important inoculum source is in the woodlots around the orchard. So this all indicates of the main ecological role of being an endophyte. And then we had this problem of these two other new species that we didn't know what they were. And once we did the sequencing of different fingerprint DNA regions, we were able to construct a family tree of these species, and we got a 100% match of the 25% of the isolates that looked with these gray colonies. They belong to a species called Choletotricum chrysophyllum. And the surprising fact was that this species has recently been described, completely new in literature in 2018 and 17, recently, infecting cashew and banana. And we do have it as a second most dominant cause of bitter rot on apple in the Hudson Valley, Pennsylvania, and Virginia. Also New Jersey and Delaware and Ohio. So that was one surprising finding. The next one was that we actually expected to see Choletotricum fructicola. That was our main expectation. And we obtained isolates from Keith Yoder's lab at Virginia Tech, and hope that we're going to find Choletotricum fructicola, because that's what we saw from the literature would be the most dominant species next to the Furiniae that I mentioned to you before, but we did not have that species. And then we had an oddball, these 12.5% of isolates that had white colonies, and they just did not match to anything that we found in this large database, which is very similar to the FBI fingerprint database to which you match the fingerprints of the criminals. So we used similar method to use the DNA sequences to match to this database, and nothing in that database matched to what we had. So then we teamed up with people from Louisiana State University, Vincent Doyle's lab, who actually put in one publication, there was just one isolate that he left undescribed, and this isolate matched perfectly to ours. And he isolated this fungus from eastern black walnut trees as an endophyte, so not causing a disease, just freely living in black walnut in Oklahoma. And we asked him to send us this isolate. Then we also talked to the Canadian guys, and they told us we were interested to sequence the whole genome of this fungus, because if it is new, it's important. So we got access through Guillaume Bilodeau to whole genome sequencing, and we teamed up also with John Gonzalez from Gillian Turgeon's lab to conduct other types of phylogeny, or basically these, doing these other types of family trees to really prove which species we have in general. And I was a strong believer we have a new species, which was not described. And it took me some time to persuade also Philip from Kerry Peters' lab that he needs to refine his phylogeny to actually see whether this is also true with his isolates. He was quite resistant at the beginning, but then eventually he also, realized that he probably has something that is not actually known before in literature. So this is just a snippet of the family tree that shows evolutionary relationships between different species. And this actually is basically a family tree of these species from the Gliosportes species complex. And we actually now are certain that we have a new species, which is called Choletotricum novaburcensis. So we named this species, and novaburcensis actually derives from a Latin translation of New York in Latin, meaning of New York. So this species is named based on New York. So we had a consensus decision on that one. We used 8 different loci, or basically DNA fingerprint regions, to identify the species. And you can see here when it clusters in this family tree, it is a sister species to Choletotricum fructicola, very close to it, and then also Nufaricola, and very close to the Choletotricum chrysophyllum that we told you was the second other species most dominant causing bitter rot on apples. So this is how this species looks like. You can see here the colonies, the Conidia, Conidiophores, Hypersoriums, and also perfect stage of the fungus, or the sexual stage, which are the asci and ascospores. So it can form a perfect stage or a sexual stage, but we don't know whether this occurs in nature in general. So why does this matter? Well, when you look at the big picture of the geographical distribution of these species, you can see these pie charts for each location where we sampled fruit. And you can see based on the color code in these pie charts for each location, you can see that the pink color is quite dominant when you go more west of the Appalachian mountain range. And when you go closer to the coast, you see a mixed presence of the blue color, which are the species that we described, new species from the Gliosporidae species complex that are more prevalent in warmer regions. So that kind of makes sense because when you go on higher elevations, the temperature optimums are pretty narrow, and that's why you see more pink for Colototricum furinia, as I said, west of the Appalachian mountain range. So we definitely believe that these species are present, based on their temperature requirements, but there are other factors also that allow these different species to be dispersed in different ways. Could you please mute your microphones, please, so that I can actually, not have the background noise, distract me. Thank you. So why does it matter also for you growers? Why does this matter to know the species? So this is a general life cycle of the Colototricum furinia. So basically, this is the cycle that we kind of renamed infection cycle, which shows you that the main infection sources can be forests and weeds in the orchards. And then once the pathogen actually spreads to fruit, it can infect fruit, and it can cycle within an orchard pretty intensively, and then eventually also be present in the leaves, in the apple orchard, not causing the disease on the leaves, being exposed to fungicides, and eventually can overwinter both in the forest and in the orchards, and spread from there into the orchards and also within the orchard. So this is just a simplified cycle. You can actually read more into it, by reading a Food Quarterly article where we actually presented this life cycle. So why does it also matter that we have different species? Well, we tested their infectiveness basically on 13 different cultivars. And you can see here on 2 different stages on the fruit, you can see that overall we have on immature apple fruit a higher, higher aggressiveness of the Colototricum chrysophyllum from the Gliosporidae species complex. So basically larger lesions that are caused by these species that are more prevalent in warmer regions in comparison to Colototricum furinia that you see on the left side. So it is important to know the species because in certain cases we can have more issues and more susceptible cultivars because of this. And we see the same result on the more mature fruit that we collected at right about the time when Honeycrisp is actually harvested. Also, what is important is that when we look at this data and correlate it to the susceptibility that is reported by growers, and this is in mid-Atlantic region, you can see these are reports of the mean % loss to bitter rot that you can see here on the left side. And you can see that I highlighted the varieties that I tested in inoculations in the lab. And you can see what is the percentage of loss for these cultivars in the different regions that the growers reported. That's what you see on the left side. On the right side, you can see the mean % loss to bitter rot from the early 2000s, and also all the way to the late 1800s. And you can see overall that loss really kind of matches pretty fine with the time when we started commercializing Honeycrisp and implementing it and planting it a lot. Which goes to say that with the increase of in acreage of Honeycrisp, there's this perfect monoculture niche that allows these fungi to really move in and create problems. So we think that one of the crucial factors that allows the bitter rot to become a larger problem in recent years is really a widespread prevalence and planting of Honeycrisp as the most susceptible cultivar. However, that's not the only other factor. There's other factors playing into it as well. In terms of the management, I've listed here which groups of fungicides we have that are really used to manage these fungi. And of course, the most effective ones are the multi-site fungicides, the M3 and M4 groups, and also QI fungicides, FRAC 11 group. However, when you look at the thiophanate methyl and DMI fungicides, basically your Inspire Super, your Rally, and other DMIs, they are not effective under high infection pressure scenarios. We know that from the previous efficacy trials, but we were mostly curious to see whether the new group of the FRAC 7 group of STHI fungicides, whether they will be effective. And we know from literature that they are variable in their effectiveness, but we wanted to see what is their effect in our conditions, and also use this new fungicide Omega fluazinum. It's a FRAC 29 group, which has been shown in preliminary research from North Carolina that it is effective against bitter rot. When we test these different fungicides in the lab, in the petri dish against these fungi, we do see a significantly different, effective concentrations for 50% suppression, in the lab. So we do see some intricate differences there in their susceptibility, but the amounts that we use in the orchard are far much higher than what we test in the lab. And it is important for us to do these tests in the lab so we can actually detect when the resistance occurs in the orchard, and how does it actually, how prevalent it is and how it changes through the time. So what we did is, to answer the question, what is effective in the orchard, we had an inoculated trial in 2020. And basically what we did is we started, spray programs on June 4th. And you can see at the bottom of this slide, what were the spray programs. So, and the way how we actually have these spray programs lined up is that basically within each spray program, either, we applied Captain, in this specific case, number 2, 8 times throughout the season, or Omega at 2 different rates, also 8 times through the season. So we can see how that one chemistry throughout the whole season can hold the fort against infection by Colotatricum furinae. So as the most dominant cause, we inoculated this plot with this pathogen only. And then we sprayed 8 times each of these chemistries. And the most interesting to us was that when we rated the disease on September 9th, we saw that overall the blue bars would show you the Honeycrisp incidence, that we didn't see much differences there. Which indicated to us that likely the drought that we had during the June and July did not favor this pathogen to really set in. But we didn't really got discouraged by this. We actually continued this trial basically and looked at it also and rated this disease on October 14th. And what we saw was that at that point, we saw a separation of the means, and we saw that overall the most effective were Captain at 3 pounds, which is your treatment number 2. The next effective were both Omega rates at 6.9 and 13.8 fluid ounces. And then out of all STHI fungicides, which are from number 5 to 10, the most effective was Aprovia, treatment number 7. And these were comparable statistically, basically as effective as Flint Extra, which is a QOI fungicide, which would be the most effective that we know. And also it was very comparable to the grower standard, which is basically was the only spray program where we had a combination of different chemistries. So why is this important? Well, because now knowing that Aprovia is effective and also Omega as well, you can actually alternate these 2 fungicides with Flint Extra and other QI fungicides that are effective, so that we can avoid overuse of QI fungicides and potentially build up a resistance to these fungicides in Colotatricum fungi that cause bitter rot. So we are going to repeat this trial in 2021 so that we can see whether these results are going to be consistent. So all of this has been published. You can access it freely. I know also you can read about this also in a new Fruit Quarterly article that is going to come in the winter issue as well. So we have a paper there. And this was a regional effort as well in collaboration to finish this project. In terms of the advices that I have for management of the bitter rot and resistance to, avoiding resistance to QI fungicides. First of all, definitely I believe that it's important to use Mancozeb early in the spring and then switch to Captain during the summer to honor the 77 days pre-harvest interval. I would advise you to use DMI fungicides to control powdery mildew and, primarily, so not use QI fungicides, so FRAC 11 to control powdery mildew, but lean more on the DMI fungicides such as Rally, Rhyme, Indar, and Procure. And never skip your pink and petalful spray. If you had issues with powdery mildew last year, then you can start an application of fungicides a growth stage earlier than pink, so tight cluster, pink, and petal fall, and use DMI fungicides to make sure you control powdery mildew well. So basically, we should definitely limit use of QI fungicides or FRAC 11 group, such as Flint Extra, Solvran, and Merivon, to 4 applications per season. And I know that that's kind of tough to do, but we should reserve them to control bitter rot during the summer, so not use them early in the season against powdery mildew. And these QI fungicides, when you apply them, you should try to use them really just before the rain that is happening after a block of hot summer days. And these are the 8 different combinations, and also you can use Captain alone, which is number 9 that I outlined here, which I would recommend you to use. So you can use Captain in combined with DMI fungicide Inspire Super or Profite early on, for the maybe second or the third cover or fourth cover or fifth cover, because these are not as effective for bitter rot control. And when the infection pressure is not high from bitter rot, usually that means before July 10th, you could pretty much get unscathed, by using these fungicides. As you get closer to July 10th, when usually that's when we start to see much higher pressure from bitter rot, you can start thinking, about using Topsin and Captan. But then once you pass the July 10th, you can actually combine Captan with Xyram, Furbam, and also, as I mentioned to you, combine it with Flint Extra or Solvran and Luna Sensation, which are standalone QI fungicides, but time them to apply them just before really rains that are following a block of very warm weather, hot weather. And you can alternate them with Omega and Aprovia now, with a high rate of Omega or Aprovia. But keep in mind that in terms of Aprovia, if you use it for control of apple scab early in the season, you can either have 5 to 4 applications per season in total of Aprovia, because that one is far much more effective for scab. Actually, it is effective both for bitter rot and scab, but you can actually maybe back off a little bit on Aprovia early in the season, use other STHI fungicides and save this one, for later. And in general, what is important is to really, use your Merivon and Pristine just before harvest, because they have 0 days pre-harvest interval. So that's your last STHI in the total of 4 that you can use, and always try to recover, just before you're going to get, the amount of 2 inches of rain. So no matter what happens, you have to recover at that time, with the fungicides. So now we have some more options, including Omega and Aprovia for bitter rot control. So now we'll switch and talk about the second section on using different molecular methods that allow us to diagnose fire blight pathogen and also quantify it at the same time. So I'll show you an example recently that we had to look at the host interactions between this pathogen with different cultivars. And this was a team effort, and basically we wanted to ask this question: Can fire blight pathogen, while present in the cankers in the orchard, can it actually activate the systemic acquired resistance? Basically, can it trigger the immunity response in bark that is very close to the cankers? And the idea behind this was that we basically inoculated a large number of trees to create cankers. And we're going to also look at the amount of bacteria that we have in the cankers. But we also wanted to see what is the response in the bark, which is the main tissue that should have some immune response to the presence of the pathogen cankers. And we kind of hypothesized that a resistant apple cultivar, if it was in optimal conditions, is going to have much higher, expression of PR genes or pathogenesis-related genes in the bark in comparison to the susceptible cultivar. And the rationale behind this experiment would be that if this is true, then we would be able to time properly different chemicals that activate systemic acquired resistance in the plant so that we can prevent formation of cankers. And what you see here in this cartoon graph is the 4 graphs on the left side, which is basically a gene expression of 4 different PR genes, okay, in Cortland, and 4 graphs on the right is in Honeycrisp. And what we did is we quantified the gene expression. And we had 2 conditions. The top 4 graphs across both cultivars were irrigated. And the bottom 4 graphs were, the same cultivars, but in the non-irrigated blocks. And what we show overall is that, when you look at the Honeycrisp on the right side in the top 2 graphs in the top right corner, we see a much more robust or significant gene expression indicating systemic acquired resistance in Honeycrisp. So for PR2, 5, and 8. And when you look at the y-axis, we see really 100-fold higher gene expression in comparison to Cortland. And so we didn't see that response in irrigated blocks, in the Cortland. And overall, when you see non-irrigated tree blocks, we didn't see a consistent gene expression, which indicates to us that it's important to have an optimal conditions for these cultivars to also, on a basic immunity level, to help control this pathogen. So along with this data, as I mentioned to you, we're going to correlate these results with the amount of bacteria that we see in the cankers, and we hope to see some differences there as well. So this is my third section. I'm going to talk about Diplocarpon cornearia now, which you might know more as Marsonina leaf and fruit blotch. And this is how this pathogen looks like. It can actually cause these spots that resemble to scab, but they do not occur at the same time. They call it scab of the summer in Asia. This disease is quite prevalent and invasive in Asia. And, they have breeding programs against this disease to create resistant germplasm in Korea and China, but not only for cultivars, but they also have to breed this resistance into the rootstocks, because when they produce rootstocks, they can get the defoliation of the liner plants. And that, is an important problem for their nursery material production. You can see here, this is a defoliation on Rome fruit trees in Medina that I pictured here. It can cause defoliation, and it did in 2018 between 40 to 60%. When you look at under the microscope, you can see the spores of this pathogen that are typical for the genus Marsonina. The spots caused by Marsonina are not the same as, as the necrotic leaf blotch. On the left side, you can see 3 leaves with necrotic leaf blotch. The middle 3 leaves that you see are actually typical spots for Marsonina leaf blotch, with some of them mixed infection with rust. They don't look really as your typical, Marsonina leaf blotch that I showed you earlier. These are the early symptoms. And you also have to, differentiate them from frog eye leaf spot, which you can see on the right side. So there is some small intricate differences. And if you would bring a sample to me, I would look under the microscope and find probably these black bodies, fruiting bodies of this fungus that would then indicate to me that it's Marsonina or it is something else. So this fungus overwinters on the leaves fallen on the ground. And basically, the pathogen likely survives in the asexual stages, conidia, on the leaf floor. We started having issues in 2018 with this pathogen because we had an unprecedented amount of rain that occurred during the summer. And this pathogen then can also form a sexual stage, which you can see up there forming apothecia, but we are not sure, and we don't know whether this pathogen forms that stage in the United States. And also that same question stands for Europe. We don't know whether this occurs and whether this pathogen forms also ascospores, which would be similar to the apple scab fungus. But that is probably a point of future research. There is a model you can use to predict this disease. When is the first infection going to occur? So the subscribers to Rimpro who work with me on 25 different farms, they can access this for free if they pay the subscription for apple scab model. It's very useful to know when the infection starts so that you can actually throw in some fungicide that's going to be very effective against Marsonina late in the summer. So this is a resource you can use, definitely. And this Rimpro cloud of models is unique by this case that it has this model. So we started from the basic questions to prove that this pathogen is causing this disease on leaves, but then we also got worried about infections in the fruit. And we started talking with Carrie Peter at Penn State, and we actually asked her whether she's seeing symptoms on fruit, and she didn't, but we did. And I'm showing you here that in Gardner, we saw symptoms on defoliated trees on fruit, from which we were able to successfully isolate this pathogen. And also we have seen symptoms on Jonagold, but on fruit that were stored. And then after a certain, in February, when the fruit were pulled out of the storage or looked at in the storage, they had spots that were, looking like Brook spot. But we isolated Marcinium from it. And then we did sequencing of the fingerprint genes that I mentioned to you was the method we identified the Colotatricum species and constructed this family tree. And pretty much all isolates regionally from New York, Virginia, and Pennsylvania, because we have collaboration with different PIs there, showed that they belong to the Clocarpum cornearia or Marcinium cornearia, 100%. So we got a great match to the database. And this disease is a newly emerging problem from New York because we did not have that in the past as a problem. So we wanted to go and look at it more in depth. So I'm showing you here the pathogenicity test. Once we got the isolates, we need to prove these fungi we have in culture are able to infect apple leaves. And you can see here how infections progress from 7 to 12 to 15 days. And you can see on the graph bar on the right, you can see that there is difference in when the symptoms actually occur when we inoculate them at the same time. There's a delay in the symptoms occurrence on Honeycrisp versus Fuji. So we definitely believe that different cultivars are different, they have different susceptibility. And data from Kerry Peters' lab indicates that's correct. So we also reproduced not only symptoms on leaves, but also on fruit. But it takes more time for the symptoms to occur on fruit once we inoculate them. And symptoms don't look anything, exciting early on, but as time progresses, they look more typical to these big black spots that would frond the edges that you see, 30 days after. So this pathogen requires a long time to cause typical symptoms, and it requires high humidity to actually to cause invisible symptoms. And that's why in 2018, that was the case. Then what we did is also we tested different fungicides in the lab against these isolates. And what we found was when we looked at these mean effective concentrations of fungicides to suppress 50% of the mycelial growth of the fungi in the petri dish, we saw that overall we see intricate differences between the isolates from New York versus the ones from Pennsylvania and Virginia. We see that there is more tolerance in fungicides in isolates from New York. But overall, the amounts of fungicides we use in the orchard for these 3 specific active ingredients we tested for, they're much higher, and all these fungicides should be effective to control this fungus in the orchard. But we do see some different levels, indicating that there's genetic background that might allow these fungi, at least isolates in New York, to potentially become resistant to these active ingredients. So we also did a gene expression assay in 2 different cultivars. So we looked at the Fuji and Honeycrisp. That's what you see here in these black bars, which is Fuji and Honeycrisp. So what we were interested in looking at is really what is the response of these 2 different cultivars, immune response to the infection on the leaves. And we looked at the genes that were basically implicated as the primary genes that indicate immunity response in the plant. And these genes basically are the precursors to this overall immunity response when there's infection by the fungus. And what we found was when you look at these purple elliptical highlighted boxes, you can see that we have saw that these PR2 genes, SERC1 and PR8, were immediately upregulated, basically expressed significantly 2 and 5 days after the infection that we did in the lab, indicating that, and this response was really most robust in Honeycrisp. So that's why you can see these red bars much higher in comparison to the black bars at 2 and 5 days. When you look at the pink ellipses that I put there, we also found that cytochrome genes and also a lysine receptor, like kinase gene, were upregulated 15 days after the infection, indicating that the immune response is likely different immediately after the infection and also 15 days after the infection. So, and this is important because it would indicate to us why do we see the difference in the symptoms occurrence between these 2 different cultivars, and also will help pinpoint what are the sources of resistance in the germplasm for the future resistance breeding to this fungus. So overall, we teamed up also with European researchers. They contacted us to give them the isolates from the United States. And this problem, started in 2010 in Europe. And it became a very serious problem because it actually defoliates trees heavily in organic orchards or low input orchards. And of course, also in cider production there, primarily Northern Italy, Switzerland, Austria, and Germany. It is so important that a lot of researchers work on it there. But in general, the epidemiology of the fungus and population genetic background is really not known. So this group that we teamed up with from Oberhansley's lab wanted to go and look at the SSR markers. And basically those are repeats in the DNA that would indicate to us also as fingerprint regions, indicate to us what, where do these isolates come from, how different they are between each other, so that when we look at the whole population, we can find out whether this was a recent introduction as a pathogen, or this was always there and then, just reappeared. So what their results indicated is that they had, these markers that looked at the different isolates showed 31 distinct genotypes of the fungus in Europe. And then by looking at our isolates, these European ones were definitely different from the 7 additional genotypes that were found in Asia and USA. So that kind of, to this group and us, indicated that Diplocarpum coronaria or Marcellina coronaria was basically having a typical signature in the population of a recently introduced species into the region. So basically had a high clonality, meaning that it was reproducing asexually very progressively, and there was very high similarity between isolates. There was a very low number of private alleles, indicating, again, a recent introduction, and there was only one dominant genotype across all the sampling sites. So in general, they also saw that these genotypes were highly distant, from the isolates in Asia and in the United States. So that was kind of supporting this hypothesis that, that this is a recent introduction from 2010 in Europe. And also there's strong evidence to say that based on this analysis, that there's a sexual reproduction occurring in Europe as well. But, these pathologists were not able to find ascospores as an indicator of the sexual reproduction so far. So there's still ongoing questions that we need to answer. And we're teaming up with different pathologists from Asia and Europe and United States, so we can answer more questions about this fungus and how important this is going to be down the road as an emerging problem. So this is Thomas. We collaborate with him. He's from an Organic Institute for Agriculture in Switzerland. And I'm going to now switch into the last section, which is the efficacy trials that we have conducted in 2020. For different problems that we have in the orchards. And before we go into the efficacy trials, I wanted to tell you that the partnership for Rimpro, outsourcing to the growers, is continuing further. This model has been very successful, and it basically, we have a core group of 25 large farms with large acreages that are interested in using this model, and they continue to be interested because it gives them really a differential advantage to know when the infection occurs and which infection actually is the one that is going to cause a problem. So this remains a large part of my extension effort. And you can see here, these are the amount of emails that I would send for prediction and interpretation of these models for the growers. You can see the amount of emails I sent for scab, fireblight, and Marcellina leaf blotch models through the emails to people who are subscribed to this model. Basically, that gives me ability to tell them the actual dates when the infections will occur. And that also serves as a basis to outsource this data to other growers outside of this group, with publishing this information in blogs without the dates. And that expands the interpretation of the models from scab, fireblight, sulblotch, and fly speck, and Marcellina leaf blotch, as well as blister spot. And also with these interpretations, I provide recommendations on which fungicides to use. And also I report when the first disease symptoms have occurred in the lab, so the growers know when to start looking for if there's any failure of fungicides. So we actually allow growers to calibrate their model by monitoring the pathogen maturity. So specifically apple scab pathogen, and we do that on 3 locations. We have done that in the last 4 seasons. We monitor scab leaf litter, and we detect when the first ascospores occur, so that these dates can be used as biofix dates in the Rimpro model. so that then the model can be calibrated correctly along with the green tip date. And that seems to be very important. And now I'm going to basically show you some data on the apple scab trial that we did this year. So on the left side, you can see the dates and the growth stages when we applied these different fungicides. And of course, the majority of them were either new DMI Sevia that is labeled in New York currently right now. We compared this fungicide with Inspire Super directly. So the dates that I highlighted there in gray are mostly the times when we applied these fungicides that are really like targeting the most, the most important time for release of ascospores during the season. We also had Stargus involved in this trial at different growth stages, which is Bacillus liquefaciens, a biological, and we mixed it with sulfur or used it alone to see how scab control is going to look like. And then Excalia as an STHI fungicide was compared in the same schedule at the same growth stages with Maris, which is a Syngenta's fungicide. And then we compared these results to see how the efficacy looks like. This is how the apple scab infection periods looked like this year. You can see there was one infection period on March 29th, which was during the basically green tip. In most commercial orchards, you would not need to apply fungicide for this infection period that you see on March 29th, because it was below the threshold basically that does not require fungicide in scab clean orchards from the last year. And then we had a battery, basically roughly 6 or 7, actually 6 of these white humps that you see, which were not infection periods. Basically, it was very cold weather. You remember probably that tight cluster lasted more than 2 weeks. And basically, this weather did not really favor these germinating ascospores, which is what these white humps represent. They were not able to establish the infection even though they were germinating. So then the only infection, major infection, happened on 30th April, which is what you can see here going above the 600 rim value, right about the pink growth stage. So if you look at this table, I've just broken down to you how these different fungicides were applied. So we early on in these spray programs labeled by numbers, we had basically contact fungicides for early stages, and then Sevia here was applied at pink, mid-bloom, and petal fall against apple scab. And then we would go back to the contact fungicides, and that Sevia was used at 4 fluid ounces and 5 fluid ounces rate, and it was directly compared with 12 fluid ounces of Inspire Super. So at the same growth stages. So that was like a direct comparison of the treatments 2, 3, and 4. And then the treatments 5 and 6 were basically consisted of Excalia, which is an new STHI fungicide indiflin. And it was basically intercepted with Inspire Super done at mid-bloom, but it was basically Excalia was applied at pink, then Inspire Super at mid-bloom, and then Excalia again at petal fall. And that was directly compared with Mirrors here in the treatment 6. And then we had Stargus applied with microtiol disperse at 20 pounds, pretty much, at green tip starting there, and then a skipped half-inch green, and then again applied from tight cluster to first cover. And that was treatment basically 7. And then we had application of Stargus at different schedules applied with Manzate or Stargus alone, and then microtiol disperse alone. So we can actually see how Stargus alone works, against this disease versus microtiol disperse alone. And then we had some other, spray schedules from other companies, including Indar and Raleigh and Luna Cessation, to see how these work. And overall, what we saw was that when you look at this graph on 3 different cultivars, we rated scab incidence on the leaves and on the fruit. You can see overall the treatments number 2, 3, 4, 5, 6, and 7 were the most successful in controlling the disease, including treatments 10 and 11. And the reason why these treatments such as 8 and 9 had less success in control of the disease is likely because they were timed differently around the infection periods. Or also in certain cases, like with Stargus here, we see that what Stargus can do alone in treatment 9 in comparison to a combination of Stargus along with sulfur, which is treatment 7. So you can see overall that largely we think that the lion's share of the effect that you see in this combination of Stargus and sulfur that is largely, because of the sulfur, impacting apple scab fungus. And then we also tested Vasiplant. I didn't mention that in the earlier slide, but we had a continuous program of Vasiplant, which is a new plant activator, plant resistance inducer, and it did show some significant effects on certain cultivars that had more genetic resistance, but it didn't perform as well as we would expect it to perform. So overall, the SDHI fungicides were very, very good tools to control apple scab, along with Sevia, which is a newly registered DMI fungicide as well. It was equally as effective as SDHI fungicides such as Excalia and Miravis. So we also had a trial with cedar apple rust control, and this was basically a comparison of different schedules of Vasiplant, Procure, and Xyrem Excel. And in the same trial, the company wanted us to go and look at the Kassumin effect on fire blight. It's quite complicated to look at two diseases at the same time, but you can see the growth stages when we applied these different products. And I'm just reminding you that in this trial, we rated both rust and fire blight, and we inoculated this trial at 80% king below. And this is the way how the spray schedules look like. We have had 5 spray programs that are listed on the left. And you can see the growth stages on the top and the dates when these growth stages occurred. And overall, what you can see is a difference within applying a DMI Procure along with Vasiplant or not at tight cluster. Then we had at pink, either just applied in certain programs Procure or Xyrem or Vasiplant. And then we had the full bloom application of streptomycin or Kassumin to compare the effect on fire blight. And then we would close the season with DMIs or Xyrem alone. And the idea was that, basically these fungicides are from different groups. If you combine them, you're going to have an additive effect. And, overall, what we saw was that you see a really good effect of treatments 1 and 3 and 5 on the control of cedar apple rust on leaves. And that's what you see on the left graph. The amount of disease you see on the leaves from these treatments. And then treatment 4 and 2 did not really work well. But then when we look at the fruit, on the right graph, you can see that only treatment 3 and 4 were the most effective. And I'll go just in a second or more, I'll go into explaining why. But we saw different responses in different organs. And then in terms of looking at the fire blight, you can see here the infection on the we also see here the effect basically on fire blight. We can see here that overall on the graph on the right here, the blossom blight, there was just no difference between the treatments overall, except treatment 1 was doing the best. It seems to us that we had either a very high inoculum level that overcame the preventive application of these antibiotics, or some other factors have caused this. And we are actually still thinking why these treatments didn't work well in controlling the disease, specifically fire blight. So I'm thinking about this, and I'm just not sure why this has happened. There's some data that indicate that potentially that there might be some other factors reducing the effectiveness of antibiotics against fire blight. So when I mentioned to you the effect on the rusts, overall, we saw that these 3 treatments had the most effective control of cedar apple rust on leaves. And the reason likely why is because in these treatments, we had Procure, which is a DMI that is very effective against rust fungi. But if you compare these 3 treatments, you can see that treatment 3 only had DMI Procure at petal fall, which indicates to us that largely the cold weather prevented rust to infect trees up until the time when the warm weather came in, and maybe only this one treatment of Procure would be enough at petal fall to control this disease. But then again, when we look at the effectiveness on fruit, so these red square boxes indicated to which treatments were effective on fruit. And likely the reason why this was the case was because we again applied Procure at petal fall in treatment 3, and then in treatment 4 we had a really heavy schedule of Xyrem, which is also very effective against rust, and that likely allowed to control quince rust on fruit in this specific case. We also had a Sudwatch and fly stick trial. I'm showing you here that we reached 190 hours of accumulated leaf wetness when the first infection starts, usually during the season, and that was around July 23rd. So you can see here we had a prolonged drought that delayed this accumulation of wetness all the way up until July 23rd. We tested Buran, which is garlic powder extract from this Canadian company, AF Global, and we combined Buran in a battery of basically 5 treatments. We combined it in 2 and 3, spray program 2 and 3 with Agrel, which is a non-ionic liquid wetting and spraying agent. And we also mixed it in a different program with Pure Spray Green and then applied Pure Spray Green alone. And we basically had 7 actually spray applications in each program of these combinations or standalone fungicides, and wanted to see what we actually get after that. And overall, what we saw was, when we look at the disease incidence at harvest, which is the left graph shows you the incidence of the disease, of the pseudobacterium fly speck at harvest, you can see that we see statistical differences in comparison to the control. But overall, the control levels were not as high as we would want them to be. So in general, this is not economically acceptable. And when we looked at the fruit 2 weeks after the harvest, when we incubated them, we saw still some significant differences, but we saw a rising incidence of the disease because the pathogen continues to progress on the fruit after there's no any more application of the fungicides. But in general, the most successful was treatment number 5 with Captain straight alone. So these results were largely similar on Macintosh as well. As I've showed you ginger gold before, and then we saw the worst results on Golden Delicious because this was the last cultivar we harvested around October 16th, and that's why you see much more incidence there. And then we basically don't see any more even statistical differences in the incubated fruit 2 weeks after the harvest. So with this, I would like to just bring to your attention that we are conducting still research on fire blight. We're actually now looking at the second year of copper affecting the pathogen in cankers. We are using copper in mixture with oils. We're trying to replicate this experiment in 2 years so we can see consistency of results. I didn't show you the data today, but we actually have done that in the past. We showed you that the data before. We're looking at the survival of this pathogen in cankers. We have inoculated trials from 2 years, and I've shown you today the gene expression, response, but we're also looking to correlate the data with the amount of bacteria in the cankers. We are looking at the copper tolerance mechanism of fire blight bacteria because the isolates we collected from grower orchards, and we have 48 of them, showed some differences in the ability to tolerate copper. And we're using some nice methods such as RNA sequencing to really key out what is the genetic basis to allow this pathogen to tolerate copper. We're looking into characterizing the strains further, and also we're conducting efficacy trials. We had issues in 2020 again with blister spot on New York 1. We're looking into the copper resistance of this bacterium, Pseudomonas aerugipathora papillans, to really key out why we have issues with this problem in New York 1. And also we're using genome sequencing to determine what is the genetic basis for this resistance. And we also have done pathogenicity and looked at the amount of this bacteria on buds of New York 1 apple cultivar. And finally, in terms of bitter rot, we're continuing this work. We're going to do another efficacy trial next year. We're doing genome sequencing on 9 strains from 4 different species that I just identified to you today are the main cause of bitter rot. And the reason why we're doing genome sequencing is because we want to learn more about the mechanisms of the resistance in these fungi to the fungicides. So we can actually learn more about those mechanisms and prevent resistance to QY fungicides from FRAC 11 group. But we're also working to look at the resistance germplasm against the bitter rot fungi. And because there is potentially a down the road ability to look at the genes that could be bred into certain varieties so we can have resistance to bitter rot. And we're looking at that now with collaborators at USDA. And with this, I would like to acknowledge different donors and also collaborators of mine and funding sources, and the people from my lab without who this work would not be possible. And I have a special thanks to the Cornell Copper Extension collaborators of mine, and also not only extension collaborators within the state, but also outside of the state. And with this, I would be very happy to answer any questions you might have. And thank you for your attention. So feel free to jump in, unmute yourself, ask questions, or you can actually look at them, and share them through the chat box, and we can talk about them.
And you have one question in the chat box from Glenn. If you can go to gene expression graphs and
Sure.
explain. She said that she couldn't see difference between Honeycrisp on the right and Cortland on the left in the gene expression graph.
Okay, sure. Let me just go and share my screen one more time. Okay, so we are looking at the gene expression for the bark, right?
I'm not quite sure if she can unmute herself. Yes, she said yes. Okay, good.
So you can see in this graph, the 4 graphs that you can see on the left side are from the Cortland. And basically what you see in the top 2 graphs, so this is across 2 years. We have 2 years of data to prove this. So you can see that in Cortland, when we irrigated the block, there was no significant upregulation of the PR genes. We basically compare this to the control, basically uninoculated trees, where there's no cankers on those trees. And we look at these red bars where the cankers are developed. And we see that statistically, there's no significant difference there in the Cortland. Whereas on the Honeycrisp, in the irrigated block, in both years, you can see these stars, like the asterisks on the top of these red bars, which indicate to you that we basically have significant upregulation of these genes. And that indicates to us that this variety has a higher higher regulation, or basically has a higher or more robust immune response, which is the systemic acquired resistance to the presence of this pathogen there. So we do see differences between the cultivars. And when you look at the y-axis, you can see that almost 100 or 120-fold higher gene expression occurs in Honeycrisp than in comparison to the Cortland. And we also have compared that to the non-irrigated blocks. We do see some numerical differences, okay, but we don't see them to be significant in comparison to the trees that are not inoculated. So, and as I said, we're hoping to see also differences in the amount of the pathogen cells, or Rhenium alvaro cells, in the cankers near this bark, which we sampled for this data set. And feel free to ask any more questions. You can unmute yourself, and if something is not clear, you can send me an email or text me. Or okay, Don, is there any other questions? Sure. Yes.
Surgeon, this is Dave Rosenberger. On that same graph that you have showing, I think you mentioned in your presentation that the gene expression upregulates when there's moisture. However, we know that if the block is not irrigated, then fire blight will shut down because growth ceases. So is the lack of, or the reduced gene expression under non-irrigated conditions really due to less activity of the pathogen, perhaps, and maybe not irrigating is desirable for control rather than irrigating to get the gene expression?
That's a great question. I think that, well, we're trying to figure out whether that's the case in a more like a pathogen-oriented way. What we are trying to do is we're going to go and look at the presence of the live cells in the cankers from the same data sets you see here. So what we hope to see is this. So this is my hypothesis, that you are right. There's less activity in the non-irrigated tree. Well, in the there's just less activity in the trees that are not irrigated. But we also think that the pathogen is stressed in these conditions, and that cells do remain alive. We don't know yet how many of them are alive or not. We're going to look at that by looking at the digital PCR numbers of the bacterial cells. But we think also that once the pathogen is stressed, it goes into this dormant state and it just doesn't replicate. It's physiologically, turning off its functions, metabolical functions. It is present there and alive, and it's just lurking and waiting for conducive conditions to occur before it can continue to maybe again replicate. So we do see late in fall, like actually early in fall, late in summer, there's in literature, it's described that pathogen can cause canker expansion when more rain comes in October and in September. So I would likely say that you're pretty much right on the money in that assertion, but we are trying to put some numbers behind it and quantify that to see. But you know, it's the interesting stuff is going to be to look at also the amount of bacteria we see in the resistant cultivar. So I'm not sure what the answer is going to be, but that's why we're doing this research.
Surgeon, you have a, I'm sorry, go Dave.
I have another question on bitter rot. I think you indicated that in the 500 or so samples that you analyzed, about 60% were Fiorini. I'm wondering if you eliminated samples from unsprayed locations, organic orchards, or perhaps poorly sprayed orchards, would the remainder of the sprayed orchards have a much higher percentage of Fiorini?
That's a great question. We did not eliminate the isolates from the abandoned or cultivated orchard. You know, that's all lump sum. So the 60% comes from 48 of the representative isolates that we used in the paper. 60% of Fiorini is out of the 48 that we identified. But when we look at all the 500 ones, the incidence was 50% of the Colopatricum Fiorini. And in the other 50% of those 492, there was these two other species. So no, we didn't segregate those. That's basically a lump sum, both from cultivated and from the orchards that were abandoned as well.
I guess the reason for asking that is my impression, and I might be wrong in this, is that in general, the C. acutatum group is more difficult to control with fungicides than the Gliosporoides group. And so I'm wondering whether under our New York conditions, New York, Pennsylvania conditions, whether the problems in most sprayed blocks really still come back to Fiorini.
Yes, that's a great question. Yeah, so I'll tell you, I've seen orchards that were cultivated in the years, a long time, you know, for a decade, and then there was one season that somehow the growers forgot, you know, to spray that block, and Fiorini was the most dominant. I would agree with you that we do see larger presence of these, but you know, we do also see sometimes that in the orchards that basically had perfect fungicide schedule. You know, then I would, you know, put my mind into having the same schedule myself. We would see sometimes no Fiorini, you know, in that orchard, cultivated, really well protected, no Fiorini in the isolates, only these two other species, like Norbersensi and Chrysophyllum. So I don't know yet how to correlate, you know, how, I'm pretty much thinking that there are factors other than the fungicides, of course, that influence when and how these species occur. While that's true that we do see a difference, you know, in more tolerance of Fiorini to fungicides, and you know, we do see much higher effectiveness of these fungicides, different fungicides for these two other species from the Gliosporoides clade. I'll tell you that Philip Martin's research in that is basically now is, you know, one is published and there's two more coming out. We do see that temperature plays a role likely in the range in which these fungi are expanding. So that's one factor. But the second most important factor, which is kind of behind what you're talking about as well, is also the number of hours of wetness hours. Basically, how long the water on the fruit and leaves is kept after the, you know, after certain dew or rain event. That's another key factor behind the temperature, and also behind the you know, behind these other factors such as fungicides that kind of shape the structure of the population. And we do see a clear trend that if you combine temperature with amount of leaf wetness hours, or you know, wetness hours in general, that you know, there's much better correlation between the incidence and also the between the incidence and the and the species presence. So yeah, I mean, I hope I answered some of your questions, but it's a tough question. I'm not sure how to, completely how to be 100% successful answering it. Kevin, did you have a question?
Yeah, I was wondering are there any like indicators for predicting bitter rot like before this season, if it's going to be a particularly bad year
Yeah.
for bitter rot?
That's a good question. So I'll tell you that there is a model, okay, that is used for anthracnose of strawberry, which is another Coleototricum species. I think it's caused by Coleototricum nymphaea. There's another species that causes that. There is if you really wish to predict it, you have to develop an epidemiological model to do that. And the tricky part is that these three species that I showed you have different temperature requirements after the temperature, I think, of 26 Celsius. After that, there's definitely, a much stronger impact of higher temperatures. Maybe it was not 26, I think it was 30 Celsius or something like that. So once you cross that temperature, there's a decline in growth of the Coleototricum furinea. And these other two species, Novibericense and Chrysophyllum, continue to grow pretty fine. So the models are not as the current models we have. You can't just be transferred directly from one system to another. And then there's just no model right now for apples that can be used. Okay, but what I usually do is I always follow the long-term or long-range forecast on a month basis. And if I see that there's going to be a trend of higher temperatures above 80. And there's going to be interspersed with that, those days are going to be interspersed with rain. I would assume that at that time we have a higher chance for infections by bitter rot. And keep in mind that this is based on historical data by Dave Rosenberger, that usually what we see in the lower and mid Hudson Valley is that the pressure from bitter rot is, really starts ramping up, right about the 10th July and on after that, onward after that. So right about that time, there's going to be more problems. So between that, between your first cover, I would say, which would be around the beginning of June, up until 10th July, you could probably apply some fungicides that are, not, excellent for bitter rot. You can, have maybe Inspire Super and Profite and stuff like that. But once you come about 10th July, you should really pay attention to those blocks of times with very hot weather, followed by a rain event, which kind of seems to really favor this disease. And irrigate early. So basically do not wait to see stress on trees and then irrigate. You have to irrigate well ahead of the stresses. So the Rimpro cloud of models that I mentioned to you for other diseases, for scab and Marcelino leaf blotch, actually has in beta mode, has a model to predict plant stress. And you can use that one as well to see on a scale of 10 days in advance when you're going to get to that point when the temperatures are probably above 80 Fahrenheit. So that can kind of, with a long-term forecast, that can aid you, assist you to make a decision when you should probably do more effective fungicides to control, kind of align them with that those critical periods for infection.
Surgeon, I'd just add that the old literature way back, I think 1903 or 1912, somewhere in there already indicated that the risk was much higher in orchards that had a previous problem and that fallen fruits seemed to contribute. So in addition to what you said, I would say that one predictor, again, the species may be changing, maybe it's not quite so true, but one predictor seems to have been previous problems. And that's why we've recommended that if you had decayed fruit, you really need to get that out from under the trees in the fall because it'll contribute to the problem next year.
Yes, I agree with that. And, there's one other piece of evidence for that is that we do see higher percentage of isolation, of successful isolation of these fungi from the buds during the winter in the blocks that had more incidents previous growing season. So I believe that when the inoculum is present in the mummies after the fruit thinning, and also on the ground as well as in the canopy, that that contributes to that whole sanitation requirement in the orchards. So by doing a good sanitation in the orchard, both on the floor and pruning out the mummies and cankers, and really kind of increases the efficacy of your fungicides. You're being a good steward to those fungicides because you're allowing them to work effectively under less infection pressure, because you're eliminating that pressure by doing a good sanitation. Yeah, I would agree with that, Dave. Yeah.
So just one follow-up question then, and it's interesting, you mentioned a higher incidence of the spores and buds where there was a previous epidemic. Do you think the copper applied at bud break would be useful in reducing the viability of those spores?
That's a great question. We don't know. I think that if the mycelium and spores are under the bud scales, I don't think that the copper is going to actually impact the fungus under the bud scales, because they're going to be protected there. But that we would have to do a research to experiments to actually see if that's the case or not.
Surgeon, you have one more follow-up question from Glenn. Then why is honeycrisp so susceptible to fire blight?
Actually, that's you should think about the you should think about susceptibility to fire blight not in really fixed terms. We don't really, the pathologists don't try to avoid discussion of that in any possible case. But I'll tell you, okay, the way how the resistance in honeycrisp is expressed. When you do, let's say you do inoculation on 6 trees, you inoculate shoots, okay, with a fire blight pathogen. And you're going to get a success between 40 to maybe 50% in success of those inoculations to form cankers. Okay, so when we talk about resistance in honeycrisp, there is different tissue resistance, that you have to consider. When you talk about the flowers, they're equally susceptible as red delicious and Cortland and Gala, because the flowers appear not to have the genetic resistance in any possible mechanical or genetic way. At least that's based on the current literature. But when you talk about the green tissue, the shoots and wood, we do see a resistance. And that the way how you see the differences, okay, is by quantifying how many trees you lose in an orchard, that is of honeycrisp versus of Gala. So there is definitely some genetic factors in the tissue. Yes, go ahead. So yeah, so that's basically that. I was going to go and say there's difference. Go ahead, Glenn. Is that you?
Well, Surgeon, I just answered Glenn's question by saying it gets more fire blight because it's honeycrisp. It gets everything.
Yeah, that's true. Yeah, I guess we can do that. Yeah. Go ahead,
Surgeon. This is Glenn.
Sure.
So you're saying that the flowers, there's no difference between cultivars for bloom infections.
No.
But there's shoot differences between susceptibility.
There is a difference in the shoot susceptibility and wood susceptibility. So so basically, we're working on a publication right now that is going to go and help quantify that better. Okay. So as I said, when you inoculate a large number of trees, when you look at the percentage of successful infections that are going to bring to formation of cankers on the tree, that's how you see why honeycrisp is more resistant. The reason why growers sometimes think there is also difference in resistance of flowers is because not all cultivars, of course, bloom at the same time. And sometimes they see infection on Gala because right about the time when it was blooming, that's where when you got favorable conditions. So that might, lead them to think, oh, well, that's why it's more susceptible because, the Gala is just genetically more susceptible. But no, if you inoculate flowers or, red delicious of honeycrisp or Gala, you're going to see, blossom blight, no problem. And, Tim Smith from Washington State University is for a long number of years when he was there working doing efficacy trials for Fireblood on Red Delicious, because he's not going to lose the trees after he does an inoculation experiment. And that's the reason how you know that these trees are more resistant. You don't lose the trees, and that becomes more important in high density orchards. Of course, along with that, with tree death, there goes also the category of rootstock. You have to choose Geneva rootstock and use Geneva rootstock, which is resistant. But if you have a resistant cultivar on a M9
Good morning, this is Larissa. I just wanted to touch base with you on a couple of things real quick.
Okay, sorry about that. I was going to go and say, the idea is that it's not only the cultivar, but you also have you have to have a resistant rootstock as well.
Is there a table somewhere that shows the cultivar shoot susceptibility differences? I'd love to see that.
Yeah, I can actually say, I'm not talking, I don't think it's a shoot resistance, table, but I think I can give you, there is a really great publication by Vanders Vet. It's called Integrated Management of Fireblade, and I can email you that. And you can see there is a table there of cultivars and how they actually clustered them based on resistance, how they rated them. So I'll do that after the conference.
Thanks.
Sure. Okay. Do we have other questions? Okay, we pretty much have to move on. I think at this point, let's go and give Donna a chance to give her talk. Donna, you can please share. Yes.
Yes, I will, but could you mute people that are still unmuted?
Yes, I'm working on that effectively as we speak. I think right now, pretty much everybody's muted, so you can share your screen and then hit the ground running, okay? Okay.
Okay. Can you see my presentation?
Yes, we can.
Okay, so once again, hello everyone. I'm going to give a overview of horticulture program and achievements at HVRL in 2020. So HORT program has been investigating the strategies for sunburn control over the last 5 years, let's say 5-6 years. I'm going to start my talk in an unusual way and open my presentation with a recapitulation of 2018 and 2019 results in order to introduce the motives for 2020 trails. So first, I'm going to talk about treatment effect on fruit quality and sunburn incidence in 2018 and 2019, and then I'll briefly cover drape net, white drape net effect on different varieties. Until now, our focus was on testing the effectiveness of the materials that contain kaolin and calcium carbonate.
Hey, Donna.
Yes.
I can't hear you really at all. It's like really muffled.
Really?
Well, now you're really clear.
Okay, I'm sorry. Okay. I guess I cover my microphone. Okay. As I said, we tested the materials that contain kaolin and calcium carbonate. These materials can form a white layer over the fruit surface and thus protect skin from direct UV radiation. This layer is also a good barrier for insects.
Donna, this is Surgeon.
Yes.
Can you turn off your browser, anything that might constrict the flow, and we might hear you better because
I don't have a browser. I don't have a browser on.
Okay, if this continues, then I would switch to my computer.
Okay, can you turn off your browser?
Yeah, I can. So just
or
anything that you have in the background.
Can you just stop?
I stopped sharing, so you're going on and you're talking. Okay, so just continue. But if this continues as a problem for us to hear you, we should switch to my computer. Okay.
Okay, can you hear me now better, or
yes.
Am I just breaking up, or you're constantly not hearing me?
There is waves at which you become less hearable, and then you become clear again, and then you become again less clear. Okay.
Okay, so that's
Okay.
All right. Can I
Continue.
just keep?
Yeah,
Yes.
sure.
Okay. So where did I? So yeah, let's just go again from this slide on. So the materials that we tested are kaolin and calcium carbonates, and these materials can block direct UV radiation and prevent heating up the fruits. So in addition to the spray products, we have tested hail nets of two colors, white and black, and their effectiveness to prevent sunburn damage. So first, let's sum up the different effects on fruit quality and sunburn incidents in 2018-2019. Although we have not found that any of the spray products can mitigate sunburn damage, we saw that kaolin-based product can actually the fruit surface temperature, which is a direct predictor for sunburn damage. When we compare hail nets with the spray products, we see that only black net can successfully control sunburn. However, that comes with a price, and that extra shading negatively reflects on fruit maturation and color development. Even in some years, yield might get reduced. For that reason, only white color has been recommended for northeastern region. Yet, white color may also negatively impact color development of certain varieties in some years. That is why we have tested the interaction between white grape net and 6 extensively grown apple varieties in the region. For this study, we use Gala, Honeycrisp.
Donna.
Yeah.
Can you speak to the screen? Because it seems that when you're closer to your screen, we can hear you better.
All right. Can you hear me now? Okay. So in this study, we use Gala, Honeycrisp, Fuji, and Empire, and two club varieties, New York 1 and New York 2, known as the Snapdragon and Ruby Frost. White drape net had a distinctive negative effect on color development of Gala, Fuji, and New York 2. It has also reduced the yield of Gala and New York 2. On the other hand, a white drape net improved the fruit color of New York 1. Honeycrisp and Empire, fruit grown under the net, show the highest resemblance to the fruit of control trees. White net had a potential to promote pre-harvest fruit drop in Empire and affect the size of fruit in Honeycrisp. So color development and pre-harvest drop seem to be the biggest concern for drape net used in some varieties. That is why we decided to focus our future investigation into drape net effect on color development and pre-harvest fruit drop. Additionally, nets have effect on yield and can change canopy microclimate, which consequently may affect the return bloom, which is of the high importance for the alternative bearing cultivars such as Honeycrisp. Since we haven't found that particles film, kaolin and calcium carbonate had limited, they actually didn't have a ability to control sunburn incidence and had a limited effect on sunburn incidence, and might require more effort to be removed from the fruit surface when packing, we decided to test products with a different mode of action for sunburn prevention. So our goals for 2020 was to investigate whether the postponed harvest of drape net covered with Honeycrisp can improve fruit color development. Also, can spray products Rain-X Plus, Parka, Vapor Guard control sunburn incidence in Honeycrisp and snow sweet cultivars under Hudson Valley growing conditions, and how the additional shading and altered canopy temperature caused by drape net affect June drop and pre-harvest fruit drop. And finally, we tested if drape net can impair return bloom in Honeycrisp. But however, since we are still working on data collection on this part of the experiment, I will not present the results at this point. So here, you can see the materials that we tested in 2020. Rain-X Plus has been tested before in our trial, but with a different rate. Since it's a wax-based product that reduces the amount of UV and visible light that reaches the fruit surface, we use it to compare it with Parka. Parka is a new product composed of phospholipids, which are supplements to the cuticle development. And we have a third material, which is a VaporGuard, composed of D1P methane that serves as a physical barrier to water vapor loss. And we also had a set of drape net treatments in white color. Here you can see the rates and dates when our spray materials have been applied, and the date the nets were installed as well. Nets in treatment number 4 were removed on the same day the control trees were harvested. In treatment number 5, we removed nets approximately 7 days later, and in treatment number 6, we removed nets 10 to 14 days later. So this WHAH stands for weeks after the initial harvest date. So now we are moving on the slides that from which you can see the fluctuation of the maximum daily temperature during the season. Blue lines present daily precipitation, and the horizontal green line is the critical temperature of 30 Celsius. 30 Celsius has been used as a general guide to signal that the air temperature is high enough to induce sunburn damage on the fruit. Our experiment was organized as a complete randomized block design. We have 9 blocks of Honeycrisp trees and 3 blocks of Snowsweet, and they are all trained to tall spindle. And this graph is just the addition to the previous slide, showing growing degree days and explaining that the 2020 was a very warm season with a growing degree accumulation of 25 to 300 degrees at the beginning of the September to the beginning of October. Not the hottest year on record, but certainly beyond the normal. Now we can move to the results section. Honeycrisp was picked on September 11th, Snowsweet was picked on September 25th, and drape net treatments were picked approximately 1 and 2 weeks after the initial harvest date. We see that Honeycrisp yield per tree was reduced in white drape net, which was picked 2 weeks later. This is probably due to the treatment effect on number of fruits per tree, increased pre-harvest drop, and to some extent to the reduced fruit size. Other treatments that promoted pre-harvest drop were a VaporGuard, drape net white, and drape net white that was harvested one week later. Similar effect of the treatments on yield or fruit drop hasn't been found in Snowsweet. However, what was constant in both cultivars is that the treatment effect on fruit size, and we have almost no exception with treatments. They are all resulted with a significantly bigger fruit size than control fruit. So on this slide, you are looking at the apple fruit chemistry. Standard traits we observe every year are fruit firmness, then sugar, described as soluble solids concentration. And we also look at the titer table acidities, and they are expressed as a gram of malic acid in 100 mils. As you can notice from the graph, none of the treatments significantly differ from the control in any of the observed parameters. This means that the sampled apples had similar maturation rate as control. However, we should consider all pre-harvest dropped apples, especially in VaporGuard and all white drape net treatments, which have not been included in the fruit chemistry testing, but were certainly in advanced ripening stage than control. So before I start commenting results about the color, I have to explain the parameters we are measured. So first we see hue. The hue tells us about the color brightness. The less negative number is, the brighter is the color. When you see the bigger negative number in hue column, that means that the color is darker. And then you have this label A with the asterisk, which is red-green coordinate, and B with the asterisk, which is yellow-blue coordinate. And A-B ratio describes the redness or greenness proportion. Practically, the higher number, the redder the fruit is. And blush is a percentage of the fruit surface covered with red. So let's discuss the results. In Honeycrisp, Rainox Plus, VaporGuard, and White Drape Net late harvest resulted in deeper and redder color of the sun-exposed side of the fruit. However, untreated control had equal or in some cases darker and redder color on the shaded side of the fruit. Larger percentage of blush covering the fruit surface was achieved with the postponed harvest of white drape net treatments. So to answer our first question, can the postponed harvest of drape net cover Honeycrisp improve fruit color development? The answer is simply yes, it can. Now we will move to the treatment effect on sunburn incidence. But before I do that, I will go over all types of sunburn symptoms and explain conditions under each of them can occur. So on this slide, you can see necrosis, and necrosis is caused only by heat. When the fruit surface temperature reaches 52 and above, it's Celsius, and this temperature has to be achieved for 10 minutes. So affected cells died and necrotic patches may appear. Next one is browning, in which affected cells do not die and damage initially appears superficial. Fruit surface temperature that will cause sunburn browning has to be above 46 Celsius for 1 hour, and this critical temperature is variety specific. Unlike sunburn necrosis, here the infrared and visible light contribute damaging skin, which becomes yellowish or brown. And the last type of sunburn damage is photooxidative, which is only temperature temperature independent. So it depends only on the sun. It occurs when shaded apples are moved into strong direct sunlight. Often the sun-exposed patch of skin will become white bleach, indicating that skin cells have died. Usually after a few days, the patch can gradually become brown, and then it can turn black or necrotic. And among all three of these symptoms, sunburn browning is the most common because it is correlated with fruit surface temperature. We measure fruit surface temperature in each of our treatments to see which of them can reduce it and thus better control sunburn incidence. So the measurements were taken on a hot day in August 11, when the air temperature was above 30 Celsius, and we had another measurements when the temperature stayed in 20s, and that was in August 31st. Both time we measured fruit surface temperature on fully exposed fruits on east and west side of the canopy. So when we analyzed fruit surface temperature, we used this statistical tool to see not only which treatment can reduce fruit surface temperature, but also to see how the fruit position in the canopy and the orientation of the row affects fruit surface temperature. You can also see what the air temperature was on the day and during the hours the fruit surface temperature was measured. And this can give you an insight on the variation between air and fruit surface temperature. The difference depends on many factors, among which are the duration of the period during the which the fruit has been exposed to the direct sun, and also it depends on the angle under which light hits the fruit surface, and depends on water stress, whether the stomatas are closed or open, the presence of wind, stage of the fruit development, etc. And as I mentioned before, we took these measurements during a hot day and during a cool day in August. On a hot day, treatments could not reduce fruit surface temperature. However, we noticed that VaporGuard, which is a transpiration suppressant, managed to keep fruit surface temperature under the level of untreated control and other treatments. Another interesting fact we discovered, and it might sound as counterintuitive, was that the direct exposed fruit in the lower canopy tends to have higher fruit surface temperature. On both days, we see that fruit on east side will receive most heat. This might be correlated with the timing the measurements were taken, and which was late morning, early afternoon. Here we can see the treatment effects on sunburn incidence. The first thing that comes to the attention is that overall level of sunburn damage for the hot and dry seasons such as 2020 was only 10 to 12% in Honeycrisp. And it really looks mild, something that we would see in an average year. Also, we see no significant difference between control and the treatments. Even more, if we look only at the numerical values, even treatment, every actually treatment except Parka had higher sunburn incidence than control. Unlike any previous year, we have not noticed sunburn necrosis, and that is why we have not reported that type of sunburn here. Again, the most prevalent type of sunburn symptoms was sunburn browning. And now moving to snow sweet. Sunburn incidence. This variety was first time tested last season and included in the sunburn trial. And what was easy to notice is that the snow sweet is 3 times more sensitive to heat stress than Honeycrisp. Again, here we see no treatment effect on sunburn incidence. However, what I want to point here is that fruit surface temperature correlates with the sunburn development. When we measure fruit surface temperature on the snow sweet, we notice that vapor guard managed to reduce it at some degree. From the sunburn incidence table, we see that although it was not statistically significant, vapor guard had the lowest total sunburn incidence. So after all, was 2020 favorable for sunburn incidence? Not more than any average season. For sun browning, fruit surface temperature has to be 46 to 49 for 1 hour. Fruit surface temperature of Honeycrisp can exceed the air temperature by 5 to 15 Celsius degree, and most likely fruit surface temperature reached 46 Celsius for 2 days when the air temperature was 35 Celsius or above. This is the reason why we saw only 10% of sunburn incidence in Honeycrisp in 2020. 2020 had actually a lot of days with 30 Celsius degree, but very few with 35 Celsius or more, and that could cause 35 or more actually can cause a more significant problem, and for instance, induce more sunburn necrosis. So to answer our second question, can spray products, Rainox Plus, Parka, and Vapor Guard control sunburn incidence in Honeycrisp and Snow Sweet cultivars under Hudson Valley growing conditions for 2020 season? The answer is no, they couldn't. Now we will move to the third section of this presentation and see how the additional shading and altered canopy temperature caused by hail nets can affect June drop and pre-harvest fruit drop. To answer that question, we had to measure canopy temperature with eye buttons. We measure fruit surface temperature with infrared gun, and we measure light interception with a septometer and observe June drop and pre-harvest drop. In the table on the left, you can see vertical distribution of the maximum daily temperature within the canopy. In untreated control and black drape net, heat tends to accumulate in the upper canopy. In contrast to that, white drape net accumulated heat in lower portion of the canopy. If you prefer graphs over tables, you can see a maximum daily temperature fluctuation over the season at a canopy level in the graph on the right. Also, when we compare the same canopy level across the treatments, we see that upper untreated control was for a couple of degrees warmer than upper canopy of black or a white net. On the other hand, a lower canopy of white net seems to be warmer for a couple of degrees than a lower canopy of untreated control or black nets. Now we are moving to the fruit surface temperature, and we can see that fruit grown under the black net had constantly lower fruit surface temperature in comparison to the untreated control or white net. Also, we see that canopy position and row orientation can influence fruit surface temperature. So the fruit position in a lower in the canopy or on the vestex position was with a lower fruit surface temperature and thus less prone to the sunburn damage. And on this graph, we measure photosynthetic active radiation. The abbreviation PAR stands for that. And photosynthetic active radiation was measured with a septometer on 2 occasions, August 1st and August 14. Again, we see the treatment and canopy position can influence the share of photosynthetic active radiation intercepted by the canopy. In general, drape net white receive 5% more light than untreated control and 10% more light than black net. Upper canopy can receive 50, middle 13, and lower canopy 7% of available photosynthetic active radiation. As I mentioned earlier, we observed fruit and counted many times during the season to be able to accurately present June drop and pre-harvest fruit drop. And what we found is that protective netting did not have an influence on either June drop or pre-harvest fruit drop. The numbers you see in the table are not the absolute values, they are percentages of the total fruit number counted per trees. Although we see no treatment effect, we can notice an existing tendency that the white net had higher pre-harvest drop and black net had smaller pre-harvest drop. So instead of looking at the treatment effect, we plotted fruit drop against fruit surface temperature, canopy temperature, and intercepted light to see if there's any significant correlation. And there were. We got a nice R-square of 0.9 for correlations between pre-harvest drop and all 3 factors, fruit surface temperature, canopy temperature, and intercepted light. However, there were no significant correlation between those factors and the percentage of June drop. And to answer our third question, how the additional shading and outer canopy temperature caused by drape net affect June drop and pre-harvest drop, is that the canopy microclimate does not correlate with the June drop, but increased light canopy temperature and fruit surface temperature promote pre-harvest drop. And to conclude, the 2020 was unordinary for many reasons. First, we had a limited access to the lab for most of the season. Then growing season started with a late frost during the king bloom, which fortunately didn't affect Honeycrisp yield, but unfortunately had a tremendous effect on snow sweet yield. Then hot and dry summer months were followed, and we had a high insect pressure, and we had a notable fruit damage caused by rodents. However, after all, we managed to bring the started projects to the end. And what we learned at the end is that the postponed harvest of drape net covered with Honeycrisp can improve fruit color development. However, the more harvest has been postponed, the more fruit will drop due to intense light and high temperature under the nets. Also, spray products Renox Plus, Parka, Vapor Guard did not control sunburn incidence in Honeycrisp and Snow Sweet in 2020. It is encouraging to see that Vapor Guard may reduce fruit surface temperature to some extent. And finally, the additional shading and outer canopy temperature caused by drape net does not affect June drop. However, higher light penetration into the canopy and higher fruit surface temperature, both are found in white drape net, can promote pre-harvest drop.
And at the end, I just want to thank you for your attention. I also want to acknowledge sponsors of this trial, ARDP, TSF, DrapeNet North America, and Cultiva. And I also want to thank my technical support in the lab, Chris and Megan. And for this, I can take, after this, I can take any your question. Thank you.
Okay, great. Thank you, Dana. So I was wondering if there's any questions, and let's look at the chat box as well. It looks like everybody's hungry.
Okay, yeah, I see one question from Robert Michael. Have you used the green net as well? No, I haven't, because the DrapeNet North America had only white and black color available for testing, but it would really nice to include other colors as well.
Glenn Kohler says, I don't understand, how does DrapeNet increase light penetration?
Okay, so yeah, numerically yes, statistically no. So last season we measured the same parameters. We see a slight reduction of the light under the white drape nets. This season when we measure, we don't see that white drape nets statistically differ from the untreated control. However, we see the difference between two types, two colors of nets. So white is definitely receives more lights than black.
So Rob Krasfeller from Pennsylvania State University is asking, did you see any longer shoot growth under the black drape net?
No, but we see huge aphids pressure under both type of nets.
And I assume you did use insecticide to control those, right?
We did, yeah, all trees were sprayed.
Okay. So, okay, do we have more questions? Well, I guess that makes it to the end. I assume if there's any burning questions, you can send them by email to both Dana and me. We thank you for your time. Attending the Apple Forum. Feel free to contact us anytime through the cell phone or email, and we can discuss any of these slides. We hope to post this video as well on the blogs of ours that we have. And if you have any other questions, there's maybe a couple of minutes more we can stay on and talk about it. But if not, we can just end the session. And once again, thank you so much for attending. Thank you, Glenn, as well. And nice seeing you through the Zoom. All the best. All the best. Bye.
Bye.